Hello guys, in this part I want to talk about blood groups and blood transfusion. On the surface of our red blood cells or erythrocytes, we have different types of antigens, which are composed of glycoproteins and glycolipids, and we call them agglutinogen. Based on the presence or absence of various antigen, the blood is categorized into different blood types. Within a given blood sample, we have more than one blood types. There are at least 25 blood groups and more than 100 antigen, which we have them on the surface of red blood cells. Because these antigens are genetically controlled, blood type may vary between them. But we want to discuss only two of these groups. One of them is ABO and the other one is RH. And you need to know that this additional RH group is present in about 85% of the population. The ABO blood group is based on two antigens called A and B. In this picture, you can see four different erythrocytes. In the first erythrocyte, on the surface, you have A antigen. When we have A antigen on the surface of red blood cell, the blood type is A. In the second picture, you have the erythrocyte with B antigen. When we have B antigen on the surface of red blood cell, the blood type becomes B. In the third picture, you have A and B antigen on the surface of red blood cell. You have both antigen, so the blood type becomes AB. And in the last picture, you don't have any antigen on the surface of red blood cell. So the blood type becomes O. In addition to antigen on the surface of red blood cells, our plasma usually contains antibodies, which we call them agglutinins. These agglutinins can react with A antigen and B antigen if we mix them together. These are anti-A antibody which can do reaction with A antigen and anti-B antibody which can do reaction with B antigen. The antibodies present in each of the four ABO blood types are shown in this picture. In this picture, you can see the blood type A. On the surface of red blood cells, we have A antigen. And in this blood, we have anti-B antibody. It means that if we give A group B blood, we have a reaction. So in the blood type A, we have the antibody, which we call it anti-B. It can fight with the erythrocyte with B antigen. In the blood type B, we have B antigen on the surface of red blood cell. And in the plasma of this person, we have anti-A antibody. So if we are B and we receive A blood, this anti-A antibody can react and can show reaction and fight with A group. In the third person, as you see here on the surface of red blood cell, we have both A and B antigen. So the blood type becomes AB. When the blood type is AB, we don't have any antibody in the blood plasma. And in the last person, as you see here, we don't have any antigen on the surface of red blood cell, not A and not B antigen. So in the plasma, we have two antibody, anti-A and anti-B. Pay attention, we don't have anti-AB. We have anti-A separately and anti-B separately in this blood type. The other blood type is called Rh blood group. The Rh blood group is so named because the Rh antigen was discovered in the blood of rhesus monkey. Because of rhesus, we call it Rh. People whose RBCs have Rh antigen or D antigen on the surface of their red blood cell, their Rh become positive. And the person who doesn't have antigen Rh or antigen D on the surface of red blood cell, the Rh become negative. So we have two types of Rh. Rh positive, it means that we have Rh factor or D antigen on the surface of red blood cell, and we have Rh negative when we don't have any Rh factor or D antigen on the surface of red blood cell. You need to know that normally the plasma doesn't contain anti-Rh antibody. If the Rh negative person receives the Rh positive blood transfusion, the immune system starts to make anti-Rh antibodies that will remain in the blood. If a second transfusion of Rh positive blood happen, the previously formed anti-Rh antibody will cause agglutination and make hemolysis, which I will talk about it later for you. So for Rh, we have Rh positive and Rh negative. Normally, we don't have any antibody against Rh in the blood of Rh positive and Rh negative, except the Rh negative person is exposed to Rh positive. In the first blood transfusion, we don't have any problem, but in the second transfusion, we may have problem because the immune system of the recipient starts to make antibody against Rh, and in the second transfusion, it can cause problem. ABO group and Rh type are reported together. So if a person has three antigen like A antigen, B antigen, and D antigen on the surface of red blood cell, the blood type of this person become AB positive. Despite the differences in RBC antigens, blood is the most easily shared of human tissue, saving many thousands of lives each year by transfusion. A transfusion is a transfer of whole blood or blood component from a person to the other person. A transfusion is most often given to alternative anemia to increase blood volume, to improve immunity. However, the normal component of one person's RBC plasma membrane can trigger a damaging antigen antibody response in the blood transfusion of the recipient. So, if we have incompatible blood transfusion, we may have some reaction in the body of recipient. This reaction is called agglutination. Pay attention. The term of agglutination is different from clotting. Clotting happens when the blood come out of the body and out of the blood vessels, which I will talk about it in the other video for you. But agglutination is the clumping, is gathering and combination of the red blood cells and antibody, and I will show you in the next picture. In this picture, you can see the correct and incorrect blood transfusion. The blood type becomes clinically important when a patient needs a blood transfusion. When the blood type between donor and recipient is the same, we don't have any problem. Look at the first picture, the first row. Here you see that our donor has blood type A, and in the body of our recipient is blood type A. So they are the same, and in the plasma of both of them we have anti-B antibody. So nothing happened in this blood transfusion, and we don't have any clumping and any agglutination. But look at the next row. Here you see that the blood type of donor is A, but the blood type of the recipient is B. So in the B blood type, we have anti-A antibody. So this antibody starts to react with blood type A. And as you see here, the antibody is Y-shaped, and by these two parts, it can connect to two red blood cells. And clumping and agglutination of red blood cell can happen, as you see here. These clumped erythrocytes can block the blood vessels, as you see in the next picture. This is the agglutination, and you can see this clumping, and it prevents the normal circulation of the blood. Eventually, some or all of the clumped erythrocytes may rupture, and they make a process which we call it hemolysis. The release of erythrocyte content and fragment in the blood is called hemolysis, and it can make the other problem, specifically kidney problem and kidney disorders. Therefore, this compatibility which we need to have between recipient and donor is very, very important. Now, in order to determine a person's blood type, we need to do typing and cross-matching. For this typing and cross-matching, we should have a drop of blood which we mix it with antiserum, and we should wait for agglutination process, which I will show you in the next picture. So during the ABO blood typing procedure, single droplet of blood, as you see here, are mixed with two different antiserum. One of them is anti-A and one of them is anti-B. So you have a glass slide here. You put 3 droplets of blood, and one of them is untreated. The second one is treated with anti-A, and the third one is treated with anti-B. And we wait for agglutination. Now you can see these different happening which happen by different blood types. One drop of blood is mixed with anti-A serum which contain anti-A antibody that will agglutinate red blood cells which possess A antigen. Another drop is mixed with anti-B serum which contain anti-B antibody that agglutinate red blood cells that possess B antigen. If the red blood cell start to agglutinate only when we mix it with anti-A serum, the blood type is A. If the red blood cell start to show agglutination with anti-B serum, the blood type become B. If our red blood cells show agglutination with both of the antibodies, anti-A and anti-B, the blood type become AB. And if we cannot see any agglutination, the blood type become O. So these are the differences between the different blood types. So we need to mix a droplet of blood with different antiserum, and then we can see the reactions. After agglutination, complement molecule start to tear the plasma membrane of donated red blood cells, and it may produce hemolysis if we don't do matching. At this time, the hemoglobin release in the bloodstream, and it blocks the filtration membrane of the kidney, and it may make many damages to our kidneys. So to avoid Blood type mismatches, laboratory technicians type the patient blood and then either cross-match it to potential donor blood and screen it for the presence of antibody. Once the patient's blood type is known, donor blood of the same ABO and Rh type is selected. In a cross-matching, the possible donor RBCs are mixed with the recipient serum. If agglutination doesn't occur, the recipient doesn't have antibody that will attack the donor red blood cells. Alternatively, the recipient serum can be screened against a test panel of red blood cells having antigen known to cause blood transfusion reaction to detect any antibody that may be present in the body. And we have a disease which we call it hemolytic disease of newborn. If we have mother with Rh negative and she become pregnant with Rh positive fetus, as I told you before, in Rh negative mother, we don't have any antibody against Rh. But at the top placenta here, we have some exposures of these red blood cell from the fetus to the body of mother. So the body of mother start to make anti-Rh antibody. In the first pregnancy, nothing happened. But in the body of mother, the anti-Rh antibodies are created. And in the second pregnancy, if the fetus is again positive, this antibody can fight with these red blood cells and agglutination may take place. This agglutination may cause hemolysis of the blood of newborn. Because of that, this disease is called hemolytic disease of newborn. And sometimes we can see something like jaundice in the body of newborn because when the red blood cells become hemolysis, the bilirubin is released, and this bilirubin come to the skin, and it make the color of the skin become a yellow. For prevention of this disease, we need to give the pregnant Rh negative women a specific serum or immunoglobulins, which we call it ROGAM. We can inject these immunoglobulins in the week of 28 to 32 of the pregnancy, and by this injection we can prevent any problems for our fetus. And the information for this hemolytic disease of newborn is here. At birth, a small amount of fetal blood leak into the maternal circulation. If the baby is Rh positive and the mother is Rh negative, she will develop antibodies to the Rh factor. And during the next pregnancy with Rh positive baby, when she transfer antibodies to the fetus, this anti-Rh antibody will attack some of the fetus red blood cells, and it can cause agglutination and hemolysis. Let me know if you have any question on this part.